Can you see my slides?
Yes.
Great. So as Anna mentioned, I'll talk to you about the GenCC, or the Gene Curation Coalition, today, and how it's a global effort to harmonize gene disease level evidence resources. And I have no financial disclosures, just to throw that out there. So I probably don't have to tell many of you, but there really is a need for curated gene disease databases just to display curated gene disease validity. I know there are a range of different databases out there, and we'll talk about how we've kind of combined them in this effort. But this is really demonstrated by this graph here on the left, where I take 14 different clinical testing panels for hypertrophic cardiomyopathy from different US clinical labs. And I plot the number of genes that are available on these testing panels, all in theory for the same indication, hypertrophic cardiomyopathy. And you can see that the number of genes varies very widely, from under 20 genes on some of the lower panels to over 100 genes on some of the higher panels. And this really points out that standards and consensus are needed for which genes are valid disease genes that are ready for clinical testing. An additional piece that we need to think of is different levels of evidence are needed for different clinical uses. For example, in predictive tests where individuals are healthy and are undergoing screening, we might want something with a very high level of gene disease evidence compared to a diagnostic panel where the prior probability that the individual is affected and might have a genetic cause might be higher. And exome genome analysis really is opening it up to all genes, all coding genes. So really just thinking about the uses for gene disease evidence in all of those cases. And the American College of Medical Genetics put out a paper in 2020 that stated for diagnostic panels that those genes with moderate clinical validity, strong or definitive, should be reported. And this is generally those curated using the ClinGen framework. And in a screening setting, so predictive tests, those gene disease relationships with strong or definitive evidence should be reported. So just to give you a sense there. So we formed the Gene Curation Coalition because we felt like there needed to be a standardized database and a standardized set of evidence level resources for gene disease validity. And so the Gene Curation Coalition combines public gene level resources, and you can see all the members for the public gene level resources here on the left, and also importantly, private gene level resources. These are clinical testing labs largely based in the US, but we have some international labs too. And so our original goals of the Gene Curation Coalition, or the GenCC, were first to understand the approaches and classification systems of the different efforts, because we know that we have different frameworks across these efforts, and we would like to learn from each other. And then, just so that we can compare our curations directly, we wanted to develop some consistent terminology for validity assessment, as well as inheritance, allelic requirement, and mechanism of disease. I'll tell you about our validity assessment terminology today. And then eventually we wanted to post gene curations from any group willing to share with the public. So our database is like a ClinVar. So this is the NCBI funded website for variant assertions. So ours is like a ClinVar, but for gene disease validity assertions. And once we had this database stood up, which we stood up in December of 2020, we then wanted to work to resolve differences and collaborate on gene curation projects. And so in order to first come to some consensus on the terminology that we wanted to use on our database website, we performed some pilot comparisons. And this is where we all curated some different gene disease pairs and presented the evidence on calls. And when we did this and compared our gene disease curations, we found that there were some reasons for the discordance, that our curations didn't match 100% of the time, and that there were some reasons for this gene curation discordance. So these are bulleted here. So sometimes our definition of disease was a little different across the resources. So maybe one group was only counting high penetrance cases, while another group was really including the spectrum of phenotypes, so lower penetrance phenotypes and high penetrance phenotypes. Another difference is the purpose of curation. For example, ClinGen does gene disease validity curations where it's just, is this gene associated with disease? We have a gene, a disease, a mode of inheritance. Whereas the panel app resources, if you're familiar with those, based in the UK and Australia, are asking the question, should a gene curation be included on a panel for a particular indication? And so those could lead to differences in the way we curate something. Also, additionally, differences in disease or phenotype assignment. Genes can be associated with more than one condition. Maybe people were curating more different conditions and saying this one is limited and this one's strong, but they're talking about different things. And then also, as we know, the date of evidence evaluation can really cause differences in gene disease evidence levels. People are publishing discovery work all the time, and much of the gene curation data that we use is from the published literature. So even if something is 2 years of a difference between curation dates, it really could be make a big difference in the level of evidence that we are evaluating. So really understanding these differences and focusing on resolving them would require harmonization of terms and also definitions. And first, we wanted to see all the types of terms that we were using. So these are some of the original members of the Gene Curation Coalition. And this table here is showing you just the range of different terms that we're using to describe the same thing. And you can see some people use strength of evidence, some people use confidence of evidence and genotype to phenotype, for example. Some people are just using the presence or the absence of the gene disease relationship in their database as validity of its gene disease relationship. And then even more confusingly, some people are using colors and they're using them in opposite ways. So the one resource was using red meaning strong gene disease relationships, and the other was using red meaning weak gene disease relationships. So very difficult to compare these things and quite confusing for the field. So this definitely proved to us that we needed to work to harmonize these terms. So what we did was we performed a Delphi survey to harmonize these clinical validity terms. Small populations and then eventually the community to really narrow down the choices for terms. For the first phase, we developed harmonized definitions that did not contain the proposed clinical validity terms, really so we were just starting on the same page. We wanted to make sure that we were all talking about the same thing so that when we came up with terms, we would have terms for these definitions. And the definitions are also present on our website along within the publication. For the second phase, we wanted to prioritize, and so we solicited as many terms as possible from all the GenCC members. And then we performed the first survey. So for this, we used the Likert scale, and GenCC members expressed their level of agreement, disagreement, or neutrality with solicited terms, and were prompted to add additional terms. And 33 people took the survey. And then for the next phase, we refined, much like you do in a Delphi survey. So we used iterative surveys. So for the second survey, we surveyed GenCC members and their curation staff. And we used a numerical scoring system assigned to the Likert scale terms. So essentially negative points for negative Likert scale terms and positive points for positive Likert scale terms and 0 for neutrality, and kind of added them together to narrow down the terms a little bit further. And then for the third survey, we surveyed the international genetics community. We received 241 responses to determine the final term list, and we included an optional 10-minute explanatory video. And we were really pleased to see that there was high consensus between the GenCC members and the international genetics community. So surveys 2 and 3 had 93% term agreement, really suggesting that we are in alignment with the larger genetics community, at least those we could survey. And so all of those terms, which I'll show you in following slides, are used to standardize submissions to the GenCC database. We also added an additional category of supportive for the less granular submissions. So supportive is generally used by OMIM and Orphanet because presence or absence in that database really is giving their evidence level of gene disease relationships, and they don't have much more granularity on that front. And just to show you some demographics from that survey, these respondents were heavily located in the UK and Europe and North and South America. And here are the populations that we surveyed with ESHG, ASHG, CCMG, ClinGen, and so on. And we asked people if they participated in consortiums, and we were actually pleased to see that the majority of individuals were not in a consortium, because we really wanted to survey people outside of consortiums and make sure that the definitions and the terms made sense. And then we asked people how familiar they were with genetics, and most people, in case we had to stratify answers, and most people were very familiar with genetics. And if not, they were pretty familiar with genetics. And when we asked people to describe themselves, and multiple responses were allowed, the most often chosen description was researcher, with also clinical genetics laboratory director or staff, and then medical genetics physician, genetic counselor, and scientific curator in the responses. And here are the finalized terms here on the right. Definitive, strong, moderate, limited. Animal model only is kind of a subcategory of no known disease relationship where there is only an animal model. No known disease relationship means there's been no identified variants in humans that are claimed to be associated with the disease. Disputed evidence and refuted evidence. So now I'm going to take some time to talk to you about the GenCC database, because really we're creating this survey to be able to harmonize all of the submissions to our database. And so this was developed and launched in December of 2020, and this is a screenshot of the summary data for now. So as of yesterday, when we had an updated submission, we have 16,911 submitted classifications on 4,704 unique genes from 12 submitters. And these are the visualization here. There's a consistent color scheme across the entire website where the positive gene disease relationships are in shades of green. Supportive is kind of in the middle because it does lend itself to evidence for a gene disease relationship, but it's not granular enough to be positive or negative. And then the limited and belows are in pinks and reds, and no known disease relationships in gray. And so those are the distribution here. And the website is search.thegencc.org. And this is the landing page here, where when you go to the main page, you'll see just a long list of genes. You can easily filter by HGNC gene symbol, or search for a submitted disease, or you can filter by submitter, and there's kind of check boxes for all of those. You'll see across the top, there are all the standardized harmonized terms that we have and the same color scheme. And you can either check or uncheck these terms if, for example, you wanted to see only definitive gene disease relationships, or those, I should say, with definitive assertions. You could uncheck everything else and just leave this, and it will filter the list for you. If you wanted to learn a little bit more about the definitions, you can just click this help button about the GenCC classifications. It'll tell you about the Delphi survey that I just told you about, and it will also give you the standardized definitions that we used. So you'll see that there are there's a line for each gene, and then you can click the little gray arrow here, and it will open up a rapid summary piece of information that you can have, where it will show you the submitters, what they have classified a gene disease relationship using the color schema, and then the condition that they have associated it with. And you'll see that there's kind of a rapid total for that over here too that you can visualize. So there's one supportive, one limited, and one disputed. If you'd like to see more information after that, you would just click the details button and it will take you to the gene level page. So the gene level page itself has the gene symbol, and there are multiple tabs so that you can sort the data by different ways. So the first one is sorted by classification. And so the stronger classifications are listed at the top. You'll see the same color scheme here. You'll see the HGNC ID with a link out if you wanted to go to the HGNC page. And then you'll see the disease here, which I'll talk a little bit more about in a second. And then you'll see the asserted mode of inheritance here, along with the corresponding HPO term if you were to mouse over it. And also, if you mouse over it, you'll see the full, so this is autosomal dominant, you'll see the full definitions or full wording for the abbreviations for the inheritance pattern. And then you'll see some dates here. The top date that you see is when it was evaluated. And then the bottom date in gray is when it was submitted to the database. So really the bolder date is kind of more what you care about because that's when they evaluated the gene disease relationship. You'll then see the submitter who submitted. If there's a link out, you can click to the public report. You can also click to their assertion criteria, or if you'd like to see more details about their particular submission, you would just click the more details button. All along the top, you can filter. So you can filter by classifications up here. You can filter by diseases. You can filter by mode of inheritance and by submitters, which might not be relevant in this gene disease relationship, but there are some gene disease relationships, particularly those associated with skeletal dysplasias, that have 20 to 30 assertions for a particular gene. So it might be worth it to filter in that case. The next tab is classification sorted by disease. And so this is where diseases are listed at the top. And just to notify everyone, the ontologies that we accept, because OMIM and Orphanet are also part of the Gene Curation Coalition, we use the monarch disease ontology because it combines both OMIM and Orphanet ontologies within it, and that's why. But for submissions, we accept Mondo, OMIM, and Orpha IDs, but all diseases are mapped to the Mondo or the monarch disease ontology. So each one that you see at the top of each of these subsets is a monarch disease ontology, and we call those disease equivalents. But we're very transparent. So for example, this is an Orphanet submission here. So they submitted as the Orphanet term, and you can link out to that. But we mapped it to the Mondo term, arterial calcification of infancy, just so that we can compare appropriately all the disease terms the best that we could. For example, this one down here was submitted as an OMIM term, and this was also mapped to a monarch disease ontology term. And then lastly, we've also sort them by submitter. So if you'd like to see for a particular submitter what they're asserting for their gene disease relationships, those are listed here. If you click the more details button for one of the submissions, you'll go to a submitter, an individual submission page. And this is often where PubMed IDs are included. You can link to their assertion criteria, so how they classify gene disease relationships. We require that every single GenCC submitter include assertion criteria in their submissions. And then sometimes they also have evidence notes. So you can read those evidence notes for a particular gene disease relationship. And then each submitter, much like they do in ClinVar, has a submission page. And so this is just the example for ClinGen. It will visualize all the classifications that they have, and there'll also be a nice list of all the classifications that you can filter for a particular submitter on their page. It will give you a little description of their resource, their website that they have, and then also the contact person for that resource and that submitter. So if you have a question, for example, about one of their curations, they know that their contact information is present publicly. So feel free to reach out and contact them. And then also additionally, a link for their assertion criteria again. And as of now, we have a range of different downloads of this data. I would say that the data are updated probably quarterly at this point in time, just to give you a sense if you would plan on putting this in your pipeline, for example. So here are the types of downloads that we have at the moment. And I'll talk a little bit about this in future slides, but we're hoping to have an API launched this year sometime. And the button for this is really just signing you up for GenCC email alerts. So when there's a big release to the database or a release of additional features, I'll often blast this email list to tell people about it. So now that we have the database up and running, we really want to start to look at the assertions for gene disease relationships and try to understand if we're conflicting for certain gene disease relationships and how to resolve those conflicts. So now I'll talk a little bit about our conflict analysis that we've done using the database. So as you can imagine, there are no standardized ways to name diseases necessarily. So each submitter has different levels of granularity for the gene and the disease that they're using. So genes are standardized, but for example, OMIM has disease ontology terms that have numbers, and those numbers correspond to the gene, for example. So someone might use an OMIM term that is the gene-specific disease term, but someone might just use the general disease term. So for example, hearing loss 1 is maybe associated with GJB2, just throwing out a random example, versus just general hearing loss. And so you can imagine without manually reviewing that, it makes it quite difficult to be able to tell computationally that those assertions are the same. So we've really tried to do different levels of granularity sorting within the database to try to identify all of the conflicts without me just manually reviewing the entire database. So we have different types of conflicts that I'll talk about here, and some of them are underestimates of probably the total number of conflicts, and some of them are overestimates of the total number of conflicts. So the first ones are the most granular ones, and they're probably an underestimate of the conflicts. But for these, we're looking at the gene, the disease term, and the mode of inheritance. And I should say that we're defining a conflict as anything that is an assertion of limited and below, so limited, disputed, refuted, no known disease relationship potentially, versus moderate and above. So moderate, strong, and definitive. And the reason that we set this is because of that original ACMG paper that I talked about from 2020 that suggests that those genes with moderate gene disease relationships should be put on a clinical testing panel. So that's kind of the line in the sand that we have, that limited and below should not go on a clinical testing panel in theory, and moderate and above should. So that's kind of a conflict if one's limited and one's moderate, or one's limited and one's definitive, just so you know how we define conflicts. So here is an example where the gene, the disease term, and the mode of inheritance all match exactly. So you know that they're asserting on the same thing. So the gene is the same. These are two submitters, just a pretend example. This is the exact same disease, the exact same mode of inheritance, but one person is asserting strong and one person is asserting limited. So we would count that as a conflict, and we would want to try and resolve that. Why is one person saying strong? Why is one person saying limited? Are they looking at the same pieces of information? Does the one missing publications, et cetera? So when we look at that, and this is data from the paper, there are 246 genes, which is 5.4% of the database at the time, that had these exact match conflicts. So we know that those are true conflicts because the disease is matching perfectly there. But how do we catch those conflicts that aren't exact matches in the disease term? Like I said, if they're just using a higher level disease term versus the exact disease term associated with the gene. So in order to try and catch that, we're looking at less granular ways to identify conflicts. So for this, we're terming it gene conflicts, where the gene matches and it's agnostic of the mode of inheritance and the disease term. And so for this, you were only looking at the gene and that someone said limited or someone said moderate. And so this is an example here where the gene's the same and the disease might be different, the mode of inheritance might be different, and there's limited and below and moderate and above. As you can imagine, this is an extreme overestimate of conflicts. Most of these probably aren't real conflicts, but at least this will flag certain entries in the database for me to manually review. So this is probably an overestimate. And this has 610 genes within it, which is 13.4% of the database. A slightly more granular way to define conflicts is gene mode of inheritance conflicts. So if we match by gene and we match by mode of inheritance and agnostic of disease term. And so the example for this is same gene, different disease, same mode of inheritance, and one is saying moderate and above, and one is saying limited and below. So this is probably closer of an estimate than this really lack of granularity gene match here. So in this case, we have 488 genes, which is 10.7% of the database for me to manually review. And so to resolve conflicts, each submitter received a report after I reviewed all of these conflicts with their conflicts, with a little bit of annotation of why they're conflicts. And just to remind everyone, limited, disputed, refuted versus moderate, strong, definitive. We actually don't count supportive in this because it's just not granular enough. And in these reports, most of the time, the most common reasons a submission was counted as a conflict is if a submission was an outlier. So for example, two submitters assert strong and one asserts limited. In these cases, the outlier would be the limited one, and we would flag that one and have them review first for the minimal amount of work. And most of the time, the outlier is often older. If the outlier is newer, though, we'll flag all the submitters and say that the submitters with the older submissions should probably review in case they've missed publications or publications have been published since they did their gene disease relationship curation. If there are only 2 conflicts, I usually flag the older one, and more complex conflicts have multiple submitters flagged, and we often discuss them on our monthly meetings. And so this is actually an example of a complex conflict that we discussed on a call, where many of these are the same disease terms. So this is the intellectual developmental disorder 59, and we've got 3 limiteds here, a definitive and a strong. So kind of all over the place, and all submitted around the same time. So we really had to have this data presented and discussed on the call. And for this, I present an overview of the gene and all the combined evidence for the curation, and each submitter preps ahead of time and can comment on their curation. And then the combined members of the GenCC will often decide on an appropriate evidence level, generally a less granular decision like moderate and above or limited and below. We don't want to tell someone you have to classify this as strong. And we offer guidance to the submitters. And submitters usually use this guidance and evidence to reevaluate their curations. And sometimes we will vote on the final classification, limited below versus moderate and above. So that's conflict resolution. And at this point in time, I would say we have resolved about 15 or so complex conflicts on calls since the implementation of the database. So to talk about future database enhancements, an API is first on the list. So we want to start with a submitter API because at this point in time, we can't ingest OMIM data. We really don't want to fork that data set and have them submitting every so often. So we are hoping to have this API in place now in the beginning of 2023. It's pretty much ready for user testing. And then eventually we would like to have a user API, but then also a login screen and a dashboard for saving searches, following genes. ClinGen already uses this login screen and dashboard. We would, because the website developer is the same, do something similar. And then because we know that people are submitting different disease terms, different granularity of disease terms, we really would like to have disease searching flexibility. So we'd like to build in the Mondo hierarchies into the database so you can have much more flexible searching. So you can search by any term in the Mondo tree, synonyms, HPO terms, and still be able to pull up appropriately the gene disease relationships that you want, for example, for panel design. And then also granular capture of mechanism of disease. We don't have that right now, but we're working on standardized terminology for the mechanism of disease with the appropriate players within the ontology world. And we're hoping to capture that in a granular fashion on the database website too. And then we are thinking about potentially a special submitter that points out candidate genes. And it's just, we call it a submitter and that it's just a dummy placeholder there, that these candidate genes would have likely pathogenic or pathogenic variants in ClinVar, and they're not present in the GenCC or OMIM yet. Just being able to point out potential candidate genes for future research. And so with that, I wanted to acknowledge the GenCC steering committee and all of the GenCC members and submitters, and then also Scott Geringer and Phil Weller for the website and database design. And with that, I will stop and take questions.